Next one, anaerobic glycolysis. Again, we take glucose, or we break down some glycogen and get glucose, but that's going to go through an enzymatic process that gives you the first step before you get into the Krebs cycle, where you're going to break this down into carbon dioxide. So all you've done is take that 6-carbon chain and broke it down into 3 carbons here. Okay, this is pyruvic acid. Then it goes to lactic acid. All you're doing is reducing that ketone down to an alcohol. That's lactic acid. Okay? Some people used to think that the pain in your muscle after the day after you work out and that sort of thing, or the burn you feel in the muscle is due to lactic acid, but I don't think that's ever really been scientifically proven. Okay? But it'll give you 2 ATPs getting to that stage. And again, it's not going to last too long. Hopefully you will be breathing within a few seconds. and the mitochondria will kick into gear and they'll start doing their thing. Okay? Because that's much more efficient than getting ATP. So this doesn't need oxygen, so it's called anaerobic. Okay? And means without. Now here's the big one. This is the important one. Aerobic cellular metabolism, or aerobic respiration for thought. This is what your mitochondria is doing. Okay? You need a combination, usually, of glycogen being broken down into glucose. Okay? That mixed with the oxygen is going to give you CO2. Okay? The oxygen actually is broken, is hydrolyzed into water. Okay? But the carbon here is coming from the glucose. Okay? So carbon dioxide and water is the byproduct, and in that process you get 38 ATPs, and it goes through the whole cycle. Okay? You can look up the Krebs cycle in the index in the book if you want to see what kind of chemical reactions are going on. I used to have to memorize those kind of things for various classes, but nowadays they don't often do that much anymore because you can just look it up. Okay? But they were all excited that they knew what it was, and so they would make us memorize it. And when you don't have glucose around, okay, so your glycogen is gone, you can actually start breaking up the fatty acids and amino acids, and they have their own little digestive pathways, remember, through the lysosomes to get into the Krebs cycle type situation. And they're going to be broken down not only into carbon dioxide, but this is where you get amines as byproducts and things like uric acid in the urine. And that's why the urine smells. Okay? You get creatinine in the urine, not creatine, but creatinine in the urine. Matter of fact, we use that to measure how well you're clearing creatinine to see how well your kidneys are functioning. So when the creatinine level starts to go up, you've got to worry about kidney function. Okay? But anyway, so the amines are broken down into other amines, and that's what gives urine its smell, and a large part of its color too. Something that doesn't mean compound. So, but real efficient, 95% of your muscle activity is going to be coming from those mitochondria. Okay. This, how does the oxygen get into your muscle cells? Well, it's attached to hemoglobin, we all know that. Okay? That's what makes your blood red. It's the heme molecule, not the protein part. You've got that big heme globin protein, okay? And then you've got a little heme molecule, a small molecule like this, with iron that's in the middle here. Okay?
Did you set up that slide?
Don't worry about it. Just remember the word myoglobin. Okay? I might have added this later before I.
Remember what?
Myoglobin.
Myoglobin. The title up here. What I'm trying to work to is hemoglobin. You know what it does. It carries oxygen in your blood, right? Okay? Well, the oxygen is bound to the iron in your blood. Okay? And it forms an iron-oxygen complex. When that hemoglobin gets close to the walls of the endothelial cells, it diffuses through. Okay? It releases and diffuses through. Oxygen basically can diffuse through like plasma membrane. Okay? Then it will be bound to this one. This is the heme molecule, very similar to what's in the blood. Okay? And what's in myoglobin. This is the protein that carries the oxygen around in your muscles. Okay? So this will release the oxygen from the iron here to the mitochondria. Okay? And it'll go back over there and grab another oxygen that's coming through. But this is how it's carried around. This compound right here, all these double bonds, act like antenna for light waves. Okay? So they absorb a lot of different frequencies of light, and that's why your blood looks red. It's because of this compound. This compound here is why muscles look red. Okay? That's where they get the color. And when they start to go bad and this starts to break down, meat goes from that bright red hamburger to the gray to the brown. Okay? But what's happening is you're breaking down this heme molecule. Okay? That's why it loses its color. A lot of the grocery stores used to kind of fake the redness. I don't know if you've ever heard that when you get carbon monoxide poisoning, that the blood looks bright red. It actually changes the way the light is absorbed in this molecule, and it looks even brighter red. Okay? So that's why you can tell when somebody has carbon monoxide poisoning, their lips look pink instead of blue. You know, they look like they've suffocated, only they're bright pink. You take a blood sample, and it looks twice as red as normal blood. Okay, that's carbon monoxide, what it does. But now they learned that you can take carbon monoxide and spray the meat with it, and the meat looks a lot redder than it should. Okay, so they were doing that in a lot of groceries. Okay. So I think most of them have stopped. I think Publix declared that they weren't doing it anymore. Okay, but it just made the meat look fresher because it was redder. But this is where the color comes from. Okay, but so all I need for you to remember is that myoglobin is the protein that carries oxygen in the muscle. One little fact. Okay. Okay, another term that your book covers here is called oxygen debt. This is fairly simple, I think, to get your hand around. Anything you're doing with exercise, I'm burning up that creatine phosphate, right? I'm burning up my glycogen stores. I'm burning up everything I have available to get this energy. And when I'm done, the oxygen debt is what is required, the amount of oxygen that has to get back in your system to sell those processes back to where they were, to build up the glycogen stores again, because remember those are enzymes that take ATP to do the reactions, and so everything I need, all the oxygen I need to get back to normal, that amount of oxygen is called the oxygen debt. Okay? And it's simple, like I said, simple to get your head around, I think. Now, myograms, these are graphic representations of muscle movement. First of all, don't confuse this with the action potential. You know, you've seen that curve. Okay, that's an electrical voltage curve that just happens to look something like this. Okay, but this is actually like a pencil drawing. If I hook a muscle from the ceiling and hook it to a beam here that has a pencil line, okay, and I start the rolling the paper, and then I shock the muscle will contract and pull my arm up, right? So as time goes by, it's going to go. I'm going to draw a trace as this paper starts to move by. Okay, that's what a myogram is. Okay? And so if I'm running the paper by and I stick that muscle, if I let it come all the way to rest, and I stimulate it again, I'm going to get a little stronger contraction the second time, and a little stronger again. Eventually, I'll reach a plateau where I don't have any increase in strength. But the reason this happens, we think, is that the first time the muscle dumped that calcium out around the sarcomere, not all of that got back in before you stimulate it again. So now you've got a little bit more calcium to let it fire the second time, and then a little bit more the third time. But also during this period, the enzymes are warming up. They're getting the temperature's actually rising, so they're being a little bit more efficient. Okay? Reactions go faster with temperature, right? So this is called the staircase effect. It's also called the Treppe effect. I have the next slide, I have that on there. And we think this is the reason why it's good to warm up your muscles before you do something. Okay? If you really want to jump that 27 feet, you know, set the world record, you want to warm those muscles up before you give it that final try. Okay? Because you want to be where the muscles are contracting at their peak, and you don't want to start out here where your muscles are cold. Okay? So that's the warm-up. But at least theoretically. Okay. This down here, don't worry about this one here. I think it's obvious what's going on. But if I stimulate a muscle, here I've let it come back completely to rest. Okay? Completely to rest each time. Okay? Then this one, I stimulate it, and before it can get back to rest, I stimulate it again. So I'm getting this sawtooth effect here. Okay? Eventually you'll get a little sawtooth, and then it'll plateau out. But this kind of curve like this is called incomplete tetanus. Tetanus is not the disease now. This is just a term we use when a muscle is completely contracted, and it's contracted as strong as it can contract, and it can't contract anymore, and it's not moving anymore. That's where you get to the peak up here. Now, but it's only a matter of how fast the stimulation is. Okay? I get incomplete tetanus if I'm not quite so fast, but if I start firing so much that the muscle just can't relax at all, then you just get this straight line up here, and then you get a spasm of the muscle where it's completely contracted. Okay? That's called tetanus, complete tetanus. So the hallmark is we stimulate so fast that there is no relaxation of the muscle on its way to complete contraction. Okay? And so I'll just maybe have one or two simple questions about this stuff. Here I put the Tues effect again, just so you have it on there. And it's that staircase pattern again. And all the rest of this is what I was just talking about as far as warming up the muscles and that sort of thing. Tues effect. Now, this is the disease, tetanus. If you're wondering what's going on, I'll be able to show you right here. Everybody is familiar with vesicles now? Okay? Here's the cell membrane of the neuron. Okay? This is the synapse down here. All right? So this vesicle is coming in. It's going around this way. We have certain proteins, and basically this is kind of PBS. I'm not going to test you on the details of this kind of thing, but I want you to see how complicated it is. Okay? You have special proteins that are in the membrane of the vesicle that were put there by the Golgi apparatus or the endoplasmic reticulum. Okay? And then you have membranes in the membrane. Here's the synapse down here. This would be the space. This is where it wants to dump the stuff out. These have to come together. This is almost like antibodies recognizing each other. This is how the vesicle knows where it should be. Okay? And it's not going to dump things out in any other place until these proteins come together. Okay? So, and this is the trigger here. When calcium bonds here, this will come down here, and this whole complex pulls the ripcord, and this membrane splits open and dumps everything out. Okay? So it's a fairly complicated little thing. This kind of stuff wasn't known when I was in school. Okay? So these are recent Nobel Prizes and PhDs that work on these things. But now we know a lot about certain disorders because, again, they're proteins, they got genes that make them. Hereditary diseases of nerve conduction can happen. Even the calcium channels that have to let the calcium in, that can cause problems with this vesicle release. Okay? So that's what's happening. This just happens to be where tetanus affects the body. Okay? The tetanus toxin destroys this mechanism in the inhibitory neurons. Okay? Most motor neurons, they call them motor neurons when they affect the muscle. There's a certain amount of tone that the brain is causing these things to fire intermittently and fairly often. You're not going to listen to a nerve with a voltmeter and see nothing happen. Usually there's a chronic tone that's going up and down the nerve. Okay? So we have to inhibit that to some extent. So we have other neurons that they're attaching to the axon or to the body of that nerve, telling it not to fire. Okay? These are inhibitory neurons. So what tetanus toxin does in the bacteria is destroy that vesicle release on the inhibitory neurons. Okay? So now the motor neurons are just firing. They're firing, and they build up a tone. The flexors are stronger than the extensors, so you pull up like this, and all the muscles in your face are uninhibited. So the zygomaticus and the buccinator and the mentalis, everything is pulling your face out like this, so you get this really weird smile, and you're all flexed up, and your back is arched. For some reason, the erector spinae is stronger than the abdominal muscles, so instead of getting a flexion here, you get that arch. I've seen pictures of old sailors, and, where the deck is full of guys that just arch like this because they all got tetanus after the battles. Okay? So let's go back. So this is not, he's not in pain or anything. That's just the way his face is being pulled. Even the orbicularis oculi there is tightened up. That's why he's squinting like that. Okay? You can see the babies are affected with the arched back here. Here's the kids. So same look on the face.